Okay, I think you're unmuted, so you can go ahead.
Am I muted? Okay, thank you. Today I will present on the reproductive biology of an globally important eucalyptus pathogen called Teratosphaera destructans. Teratosphaera destructans causes severe shoot and leaf blight, which leads to the defoliation and dieback of young eucalyptus trees cultivated outside of their native environment. Asexual fruiting structures form in the stomata under moist conditions, and they exude these asexual spores. The fungus has only been reported to cause disease in tropical and subtropical growing regions, specifically in Southeast Asia, but in 2015 it was reported to cause disease for the first time outside of Southeast Asia and in South Africa. Our previously work identified that destructans has an outcrossing mating strategy, which means that two individuals of opposite mating types are needed for sexual reproduction to occur. We then assessed the mating type distribution of a global collection of isolates and found that each population had one dominant mating type. And very importantly, our South African population only had one mating type. These findings suggested that the asexual cycle most likely represented the primary mode of reproduction for this fungus. However, very little is known regarding the origin, pathway of dispersal, or the reproductive biology. Thus, the aim of the study was to investigate the genetic diversity, structure, and potential for recombination in a global collection, with a very specific interest into the South African population. This was achieved by developing and applying 10 polymorphic microsatellite markers to the same global collection of isolates. We found 18 genotypes, of which 17 were limited to their specific population. Only one genotype was shared between some of these populations. Specifically, we had 2 isolates from North Sumatra and Indonesia, some isolates from Thailand and Vietnam, and all isolates from South Africa. In the minimum spanning network, our genotypes generally separated according to their sampling region, indicating independent evolution after the pathogen was introduced into the respective areas. These areas are geographically separated from one another. For the population structure, we used 2 methods, and both of these methods assigned our individuals into 3 clusters. Again, we can see that our isolates grouped according to their geographical location. With the 2 Indonesian populations in cluster 1, all of our populations with the shared genotype in cluster 2, and all of our Chinese isolates in cluster 3. As I previously mentioned, the South African population had one mating type, and now we can see that this population is also entirely clonal. All of our populations had low genotypic diversities, with the highest genetic diversity of only 10%. And this was found in one of our oldest known populations from Indonesia. The hypothesis of random mating was rejected for all populations, and therefore we saw no evidence of recombination. This was not really surprising, since our data set consisted of more than 89% of clones. To recap our findings, all investigated populations had low genotypic diversity, high levels of clonality, no evidence of recombination, and strong clustering among populations, which suggests that the asexual state is the dominant mode of reproduction and dispersal of this pathogen. This is consistent with the fact that only the asexual state is known and regularly found in heavily diseased trees. All of the investigated populations also represented non-native populations, which were introduced independently into the respective region. Therefore, the origin of this pathogen remains unknown. Lastly, the most recent occurrence of this pathogen was in South Africa, and it was entirely clonal. This was despite the fact that collections were from multiple sampling sites across South Africa. The pathogen most likely entered South Africa with a single introduction. I would like to thank everybody that is involved with this project, and I would like to especially thank you for listening.
Thank you very much, Minette. Lots of virtual applause. It was a really, really interesting talk. So I've no background in fungal pathogens or eucalyptus trees, and I'd like to know just some basics. So eucalyptus trees themselves are not native to South Africa, right?
No, eucalyptus is native to Australia, mostly native to Australia or a few of the Asian islands.
Ah, okay. And it was obviously brought here by any colonist or anyone particular wanting to bring a tree here. And likely they kind of brought these funguses with them.
So we brought eucalyptus mainly here for plantations because our native forests aren't producing enough wood for the timber and pulp that we require at the moment. And eucalyptus is an amazing tree for that specific purpose. And we believe that this fungus was only introduced in South Africa in 2015, so it's very recent. We believe it came with some importation with some seeds because we regularly need better quality plant material, and that is the general way how these plant pathogens move globally is with plant material.
Okay, I see. So this is a very recent pathogen then. We've got quite a few questions in the comments, so I'm not going to bore you with my plant mystery questions, but maybe if we have time, we'll come back. So we've got a question from Natasha. What does the low genetic diversity mean for the future of this fungal species?
So specifically, let's focus on South Africa. The fact that we don't have any diversity is a good thing for South Africa. We're focusing on resistant breeding for this one genotype. For low genetic diversity, it can mean anything for the rest of Southeast Asia. We have low genetic diversity, meaning that the sexual reproduction just doesn't occur that often, meaning we probably won't get a better genotype. But it's not necessarily the case. It just might mean that we haven't found it yet, or we haven't looked at all of the areas where this pathogen might occur. Maybe we get new introductions and sexual reproduction could potentially occur.
Okay, interesting. And there's a follow-up asking, is there a primary import source of eucalyptus that comes to South Africa? Would we mainly buy from one particular region?
No, we buy from Australia and Indonesia mostly. They are two of the biggest. But we are a very big eucalyptus country. So we also export a lot of our plant material. So we can also be a source for potential new outbreaks in other parts of the world.
So we could be spreading it on. Oh no.
Yeah.
Okay. We've got a question from Issa. Could you extend on the set of microsatellites that you used? Do you think that by having a larger set of microsatellites that it might impact the overall genetic diversity? And thank you for the talk.
So the first test that I did do is to see if we can get 100% of our genotypes, and we could get 100% of our genotypes with 10 microsatellites. I don't think increasing it would have changed the genetic diversity by much. It would have still been very small. We will still have a lot of clones. It is definitely a clonal pathogen in these areas specifically.
Okay, very, very cool. And very, very interesting. And then maybe just a final question to save my curiosity here. So we probably could design strategies to inhibit the fungus for farming methods. But what about invading eucalyptus? Could we weaponize this fungus?
So this fungus only occurs in tropical regions, and where we plant eucalyptus as a plantation crop for beneficial purposes, that is in tropical areas. So here in the Western Cape, I'd say it is maybe an invasive tree, but it would not be able to infect these eucalyptus trees. And so far, the species that it do infect is our plantation species that we generally use the most. So the globulus and the other species that's in the Western Cape isn't susceptible to this fungus. So I don't think so. No.
Bad luck. Anyway, thank you for a really exciting talk. I really enjoyed it.
Thank you.
And yeah, it was great to have this present here. Cool.